Thanks everybody for coming. I appreciate you coming, everybody coming out here to see this. Um, so the idea here was just to offer everybody a chance to present their the research and the project that they did this summer. We'll have 5 presentations today. I thought we could start out with the 2 presentations that were sponsored by the Michigan DNR, so John and Becca. And then we can move on to the 2 that were sponsored by the Detroit Zoological Society. But so Sarah and Kyle conducted those. And then Ethan Shays from Detroit Mercy conducted a really nice local study at a community garden. So we can wrap up with his presentation then. So yeah, so I guess we can just get started.
Hi, everyone. My name is Becky, and I go to the University of Michigan. And this summer, I was working at the Charlevoix Fisheries Research Station with the Michigan DNR. This summer, we did a bunch of different projects. We kind of tried everything, but my main points of focus for this presentation are steelhead telemetry and steelhead life cycle analysis. So before I get into kind of like the research that we did, just a bit of background information. Telemetry is the process of recording and transmitting the readings of an instrument. So in our case, we were using acoustic tags, which are basically embedded in the body of the fish and then kind of like gives back information, whether you do like an acoustic survey or like obviously for us, we just had the tags at the station, so we were able to collect information. The reason for this project is because steelhead populations have declined from 3.5 million in 2011 to just under 3 million in 2018, and this trend has continued. So these acoustic tags should give us a better idea of just kind of like steelhead movement in general and like why they're dying, what's going on with the decline. And then it will also tell us more about predation, which is something that other tags don't really have the capacity of doing. So our research was, will acoustic tags be suitable for steelhead populations? We're using steelhead smolt, and obviously they're not very big. So does the body size ratio matter, all that stuff? And then will they better help us understand the reason for population decrease? So the way that this research took place is there were 63 steelhead smolts kept at the station from April to July. So the project actually started before I got to the station and I kind of just joined in. So there were 20 with a dummy acoustic tag, 20 with a suture and no tag. So basically they had stitches where the tag would be, but we didn't actually implant them. And then there were 20 control fish, so that had nothing. Before these fish were all placed in the tank, they were measured, weighed, and pit ID'd. And then once they were tagged, they were all put in the tank with the same exact conditions and kind of like mixed up. So we had no idea which one was which. And then finally in July, I'll show you actually on the last slide. These are all the fish that we dissected, and we just dissected all 63 because we didn't know which ones had the tags in them since they were all combined. And then checked for tags, ending height, weight, and just kind of like a post-surgery analysis. So if any of them had a messed up stitch or if the dummy tag was coming out of the bottom, like that was all things that we were noting. So these are some results. This is probably the most important graph, this condition factor. As you can tell, the blue is the start. So the control group like didn't start as the strongest, but as they kept going, their condition factor went up. So they were improving in life a little bit, but the dummy tag kind of had a significant decrease down, which is a little bit concerning. And then, so this is changing condition factor. The control is going up, dummy tag and suture. Suture is staying the same and dummy tag is going down. As you can tell over here, like, yes, they all kept growing in length, but in weight, the dummy tag did not grow as much, which is also just kind of something to watch out for if they're in the future, if they're in a real water situation and they're just not growing as fast as their peers. Yeah, so these are our results. Maybe check them out for a little. And then kind of a summary is, while the condition factor of the fish with the dummy tags did have a significant decrease, we're still planning on funding this project just because it is really important information and it will tell us the why of why the populations are decreasing rather than just that they are decreasing. And this is kind of very valuable information. And also it was a decrease, but they were still able to survive among their peers. So that's kind of interesting. But basically, these tags are very specific. So they'll show when exactly in the life cycle they died, if they were eaten or caught or anything like that. And that's all kind of pretty valuable information for this project too, which is what I'm going to be continuing in the fall. So steelhead have been leaving rivers and entering the lake as 1-year-olds as opposed to 2-year-olds, which is kind of like the trend of the past. So is this contributing to a population decrease, and why are they leaving early? Like, why is this going on? So hopefully these tags will be able to tell us at least the answer to the first question, is this contributing to a population decrease? And then finally, obviously the project was a main component of this internship, but really we did a different thing every single day. And I'm so grateful for all the opportunities we had. So here's some alternative projects we got to work on. Reef restoration project, that's a huge thing going on at the Charlevoix station. We did the acoustic surveys where we went out in the middle of the night and just kind of like looked for fish, I guess. We continued Sea Grant 21 Michaela's Goby project, which is kind of her doing camera drops and stuff like that for the Goby populations. We got to try crewing. We did a bunch of surveys, so lake trout, smallmouth bass, and so much more. So these are some pictures. That's me on the steelhead in the corner over there. That's John holding a big fish. But yeah, so that was my project and my time. Thank you.
Hi, everyone. My name is John Hayhurst. I am going to be a junior at Michigan State University in the fall. I also worked at the Charlevoix Fisheries Research Station through the Michigan DNR with Becky, and worked closely with my mentor and advisor, Dave Clapp. Again, I did a lot of overall experience stuff, just like Becky did, but my main focus was growth and survival of age 0 yellow perch in eastern Lake Michigan. So we'll look into that. So just a little background. The project that I was working on is a replication of a couple studies by John Detmers, David Wall, Michael Weber, and Scott Meals. 2 different studies actually. And so in those studies, they studied age 0 perch in southwestern Lake Michigan, so in the water just north of Chicago. They examined otoliths, which are a bone in the ear of the fish that can give information about life history, daily ages, hatching distributions, that sort of thing. And then that second study was looking at factors that influence habitat shifts. So in that middle graph, it kind of shows life history of yellow perch. And so at what ages and stages do the fish move habitats? So shallower, offshore, a little deeper. And so I just put a picture of a tree. So I know a lot of you probably know that a tree grows in rings. And so you can kind of tell age and growth and how that tree grows. So in the bottom right picture is the otolith, and it kind of does the same thing. So it'll grow in rings, and with that, you can look through a microscope and tell certain information about that fish. So just a little bit more about what the project is. So why do we want to replicate this? Why do we want to spend time in it? First and foremost, just collect information about yellow perch life history. So obviously it's always important to collect data, and then we can use that data to draw parallels to other fish species. So lake trout or whitefish, different fish that are, in our watershed. And then try to indicate and predict year class strength, which is essentially how many fish hatch during a given season. And that's super important because if we can predict that and understand that, that provides information about yellow perch fisheries, quality, success in the future. And just monitoring those populations. So some of the objectives is just obviously find and compare data from the fish that we collected on Eastern Lake Michigan to those previous studies just north of Chicago, and then analyze different factors that are influencing yellow perch survival. So a big thing that we did is kind of looked at information before some invasive species came, so especially round goby. And so looking at information that we collected before the boom of the round goby and then afterwards, and see if, and those invasive species are influencing life history. And then try to identify a settling or benthic movement marker on the otolith. So basically, if we can look at an otolith from different fish and see when, if we can see on that otolith when a fish may be moved to a different habitat. Okay, so here's just kind of a flow chart of what I did. So that top picture is the lab that I worked in a lot, my little station. This next picture is the archive fish that we had. So the fish that I actually worked on, as you can see from those dates, late 90s. So almost 25 years old, those fish have been in those containers. So I looked at those fish before kind of the boom of the round goby. And then we'll look at fish afterwards. So then I pull them out, put them on my Petri dish, record length, and then actually do extraction. So that bottom right, I know it's a little hard to see, but you can see on my pointer. So I just try to get a good picture of where the otolith actually lie in the fish. So kind of at the base of the skull and the spine. And so I would go in with my tweezers and picks and pull those out, then put them on slides with their information, where they're from, with a little glue on there. And that slide is what they look like under the microscope. And then this last one is some sandpaper and mineral oil. So I'll sand those down so then we can get better pictures and viewing capability of what they look like under the microscope, which is this slide. So I kind of just tried to show what it looked like. So this first slide is I'm sanding it down a little bit, right? What it looks like. You can kind of start to see some of those rings. Sand it down more. I had to be really careful with this because if you go just a couple strokes too much, then you either lose the otolith or it breaks. Or the next slide, you can kind of tell it's a little bit lighter. And then I just increased the power on the microscope. And so you can see more and more those rings show up. We should be able to count those rings and see if we can find certain markers that helps us with that life history. So then, just like Becky talked about earlier, I spent a lot of time doing that, but I spent also a lot of time working on other projects. So I tried to take as many pictures as I could and soak in that experience. And I'm so grateful for everything that we did out there. So like Becky talked about, we did a lot with the Reef Restoration Project, round goby, abundance surveillance, which are those big tanks on the right. So we kept goby in there and did research on that, and then different surveys, much more crayfish stuff, diving stuff. And yeah, so that's all I have. Thank you.
Hello, my name is Sarah Kresman, and I am from Central Michigan University. I actually graduated this past May. And this summer I worked with Kyle at the Detroit Zoo and did some community outreach and educational programming. So at this internship, I did two main things. I created a program to educate K-12 students about the relationship between tree canopies and air pollution. And I also created a survey and worked with landscape architects to ask about multilingual signage in Southeast Michigan parks. So for the tree canopy program, we presented it at the Detroit Youth Day, and there were three main stations. At station 1, students learned like what air pollution was, which is the photo on the left. Station 2, they learned where air pollution can come from. And station 3, they were able to identify some different trees that were in their community. And there were many different activities involved, such as spot the difference, some exercise movement activities like jump rope and like a potato sack race, and then some matching activities as well. And then for the multilingual park signage program, as I mentioned, I created a survey to help gauge community interest in implementing multilingual signage in parks. And I also communicated with some landscape architects to see what they were already planning in terms of implementing multilingual signage in their communities. And the end goal of this program was mainly to kind of see what communities wanted in terms of multilingual signage and use the input that we got from like the survey to help gauge the next steps for the program. So for example, if communities decided that they wanted more multilingual signage and there wasn't that much representation in their area, I would help them work with landscape architects and park leaders to help put that into motion. However, if they found that it was not something that they necessarily needed or wanted in their community, then we would leave the parks as is. For example, one of the landscape architects I talked to, the parks that she was in charge of, they didn't really seem to need a lot of multilingual signage, but they were putting in tactile and braille signage for visually impaired community members, which I thought was really cool. But overall, I was able to also really gain more information about what Southeast Michigan parks were already doing, especially in the Detroit area in terms of multilingual signage. And the photo on the right is just a screen grab of the survey I made. So overall, this my time at this internship really helped me understand community needs of the local community better. As I mentioned before, I created a survey to help gauge community interest in multilingual signage. That really helped me kind of see what the community wanted. I also am from Southeast Michigan, so I knew that air pollution was kind of a big concern in the area. So I was able to work with community members to create a program to help educate others, especially children, about the problems with air pollution, and then some solutions that were already implemented in their community, such as how trees can help remove air pollution. And then these two photos are from the tree canopy program. The one on the right is a matching activity to help teach participants about where air pollution comes from. And the one on the right is the potato sack race. That's me and Kyle doing it that's between station 2 and 3 for the participants. But overall, I was able to research and just work with community members to see what kind of things that they wanted in their community, which helped me better when I was creating solutions and programs to implement their input and knowledge as well. And finally, I had many aha moments of like, wow, I'm really glad that I am in the right field and what I want to do for my career and stuff. And one of them was from working with the K-12 students at the Detroit Youth Day. So the Detroit Youth Day has been going on for almost 40 years now. It's usually held in Belle Isle every year. And it's when a day of like games, fun, and activities to bring communities together. So Kyle and I presented our tree canopy program here, and are able to see the students really get engaged with the different activities and learn a lot about tree canopies and air pollution. The photo on the left is actually from the tree identification station where participants can identify different trees in their community. And I was able to see the kind of direct impact my program was having, like right away, because there was some students there. One of them, he came up to us at the station and he realized that one of the trees that he identified was a tree in his backyard. So he got super excited and he was asking like, oh, like what does it need to grow? Like, can it grow anywhere? Can it grow on the beach? Like, really excited that there was already something in his backyard that was helping clean the air. So I thought it was a really great opportunity to see my program already teaching people, and that's something that they can take with them as they like, grow up. But yeah, overall, great experience. Really enjoyed my time. Any questions, Al?
So hi everyone, my name is Kyle. As you heard from Sarah, I was also part of the Detroit Zoological Society, and throughout the whole summer, we did a lot of community outreach and different programming to engage the people throughout Michigan about a bunch of different environmental concerns. But I'm from a student athlete from Eastern Michigan University, and I created and was a part of 2 different events. One that you can see there, I created an environmental stewardship event that's going to be hosted annually, and there was a collaboration amongst multiple different environmental science programs and organizations throughout Michigan. And the second one, which Sarah kind of talked about a little bit already, which was the Youth Day event. So the best way for me to describe how this summer went is to kind of go backwards in time. So here was my School of Green event. Like I mentioned earlier, it's an annual event. It was hosted the first year on July 26th from 1 p.m. to 4 p.m. at the Ann Arbor Senior Center. And each year the School of Green event happened is going to be a different animal or species focused on. So for this first year, it was Herpetofauna, so reptiles and amphibians. So everything at the event was centered around conservation and protection of our local reptiles and amphibians within our communities. And on the next slide, I can show you a little bit more. But here's kind of how the day went. So there was obviously some really good food and refreshments there. And then like an introduction of the event. And then next I brought in the Natural Area and Preservation of the City of Ann Arbor, and they talked about some of the Herpetofauna survey programs. So lots of times throughout the summer and spring, they look for volunteers to help them scan and locate different toads, amphibians, such as like salamanders and other things as well, and reptiles within the Ann Arbor region. And they kind of count for how much is in that population in that given area in that given time. So they sort of talked about their program and talked about the importance of surveying and looking at the different populations of reptiles and amphibians within the City of Ann Arbor. Next, I have a live wildlife presentation, which I had some photos to show you from that as well, where I had the Huron-Clinton Metro Parks Mobile Learning Center there, and they brought in some live animals to show the participants of the event. Because the main focus of the School of Green event is to educate people about some of the animals within their communities, as well as have people feel a little bit more comfortable with some of these animals. So they brought in a snake, they brought in a couple of toads, they brought in a salamander, and a few other little creatures as well. And people were able to touch them up in person and be able to look at them in real life. And then I had a brief speech about stewardship and the different ways we can protect the local reptiles and amphibians, such as properly disposing of nets and fishing materials when you're done with them. Also like not littering, not driving off of trails through forests and whatnot, just different practices such as that. Next, I had Sarah talk about her multilingual signage survey and kind of the importance of having inclusive programming and signage within throughout Michigan. And then we had a nature walk and a park cleanup where we went to Burns Park and picked up all the trash we found up there, which was surprisingly a lot there because it was not too long after the 4th of July. And then I had some closing remarks. So here's some photos from the event as well. This is the Natural Area Preservation and their program there. Here are some of the animals that was brought in by the Huron-Clinton Metro Parks Mobile Learning Center. And then on the bottom, you can kind of see the park cleanup when we went outside and picked up trash and whatnot. I've already started planning for next year. This event is going to be adopted by the Sierra Club at Eastern Michigan University, and many of the same different organizations I partnered with, such as the NAPS, so the Natural Area Preservation, the Give 365 volunteer program, which led the park cleanup, the Metro Parks Mobile Learning Center, and so many other different organizations have already helped me and already started planning for next year's theme and already looking to go forward with that one. So I'm really looking forward to having this event go on for years to come, and I'm really looking forward to seeing where the Sierra Club at Eastern Michigan can take with this event. Now I'm going to go a little bit into the Belle Isle Youth Day event. It happened on July 13th. We were there pretty much all day. Sadly, this year it rained, but usually there's around 35,000 kids in attendance there. Some of them are coming from summer camp, some of them are coming from school, some of them, their parents are just bringing them there, but usually there's a lot of people in attendance at this event. And there's multiple different organizations and nonprofit businesses and organizations talking about some of their either research or their business there as well. So there's kind of just a stretch of different activities for kids to be able to learn about some of the different environmental or just other topics while being able to meet some of these awesome different organizations and businesses throughout Michigan. I collaborated with Sarah on this, and at the first station, we kind of have a spot the difference. As you can see on the far right there, we had 2 pictures, and we kind of had the kids kind of guess, what do you think is the difference between the 2 pictures, and why do you think one kid, I think I named him Tommy, and the other kid in the picture, like little Jimmy or something, why do you think Tommy's happy and why do you think little Jimmy's sad? And then kind of going from there. And then the next station, which is the one Sarah was at, she talked about where some of these harmful pollutants come from and having the kids identify on this board where some of these different pollutants are coming from. And then the third station, as you see on the picture on the left, we had a tree identification game where they had names of the different native tree species within Michigan, and then they placed the names to the image, or like we had cutouts of like the leaves and branches of those specific trees, and they were able to kind of do like a mix match and match the name to the tree there. Some of the key takeaways I had this summer was definitely being able to break down some of the stigma surrounding reptiles and amphibians. A lot of people going into the event messaged me or told me in person, my School of Green event, that they were kind of nervous about having live snakes there. Some people were not a fan of that, but by the end of it, a lot of people felt a lot more comfortable. A lot of people enjoyed the event. I believe I had 51 people RSVP to the event. I had exactly 40 people attend. So considering this is over the summer and it was like an educational event, I was really happy with that because a lot of people were telling me to aim for 8 to 12. So I'm glad I knocked that out of the park. And then the next big takeaway I had from this summer was I was able to firsthand encourage young adults, people K through 12, about environmental stewardship and encouraging them that if you have a passion within the environment or sustainability or anything along those lines, you can find an interest or something along those lines in this field. Next thing I realized and kind of took away from this summer was I was able to also see the diverse field of environmental science. When I was creating my School of Green event, I worked with people from all different backgrounds of environmental science. I had environmental science engineers, I had biologists, zoologists, I had people with all sorts of different interests that just had a passion for the environment and just really wanted to help me with this STEM education programming. So they helped me with my School of Green event. They came, brought out flyers and talked about their different organizations and surveying they do. And the last big takeaway I had was that I was able to meaningfully engage with the community in a way I never had the opportunity to do. One of the big things and one of the big reasons why I chose to work with the Detroit Zoo is I wanted to create a sustainable programming, something that would go on forever and be able to impact people, not just this year, but on the years to come. So I believe I was truly able to do that with my School of Green event, as well as the Belle Isles event, since a lot of the kids really enjoyed it and felt like they really learned a lot from it. So that's all I did with my 2 events.
So we heard like 2 of the sponsored projects from the Michigan DNR and then 2 from the Detroit Zoological Society. So again, I just want to really thank the Michigan DNR and Dave Clapp and Mike Reed and the Detroit Zoological Society for providing such a great experience for our interns. They've been partners on this internship program for several years now, and we hope that continues. It's a really great experience for the students, and you can see it has impact on the communities, and then that's great research for the project. And then, so in addition to the sponsored internships, we also have student proposed projects, and those can be a variety of different projects. So we'll have 2 of the student proposed projects then.
Hello, everybody. My name is Ethan Skies. I just finished my freshman year at University of Detroit Mercy, and this is my project on using Google Earth Engine to evaluate how urban forests in Detroit impact the distribution and movement of water. And it's affectionately known as the Wamanomenon, as that is Palmer Park Forest, my community partner. So my community partner was Palmer Park. Palmer Park was sold to the city with the understanding that it would remain undeveloped. And now the trees in the forest are reaching their maximum lifespans, and we aren't seeing replacement of successive generations because of the local deer eating any new growth and the lack of gene flow, as no animal is brave enough to cross all of Detroit to bring in new genes as seeds and stuff like that. So in order to understand the forest and its benefits to the ecosystem, my project is focusing on the hydrology of this specific urban forest. Google Earth Engine is a tool that makes visualization and collection of data extremely simple once you understand how it works. So the bulk of my project and time was spent learning the capabilities of Google Earth Engine. To accomplish this, I needed to learn a new programming language, Earth Engine specific functions, and just how to manipulate the image layers properly. I took many courses and even posted to my trusty source, Stack Overflow. The more I learned, the more I added to my project. So first I needed to define the area in which I was wanting to work. So I hand placed the vertices of polygons. So as you can, all the highlighted area is hand traced forest area. And from there, I used a small bit of code that took me way too long to figure out to essentially conduct virtual transects. In previous study, my freshman year actually worked with Palmer Park as well. So this is kind of continuation of that, more focused on computer sciences. That's my area. We ran actual transects and collected elevation data. So the first image layer I tested was elevation. And in knowing the general location of those original elevation, where we collected the original elevation data from, I was able to get a rough estimate of how accurate it was. And it was off by about 1 to 3 meters, but the inaccuracy is due to the resolution of the image layer, because each pixel is the average of that location. So they average out the elevation data for that 1 pixel. I was then able to look at flow accumulation, which is showing where the flow of water is accumulating in the region. And as you can see, Palmer Park, so that pink dot in the middle is very zoomed out. It's surrounded by major watersheds. So you have the Detroit River coming in at the bottom, the Rouge River going up the side, and then Red Run coming into the top. So it's contributing a lot to the surrounding watersheds. And along with flow accumulation, I was able to look at the drainage direction, that side on the side there. And that is basically showing the water direction flowing out of Palmer Park. And my original test for the virtual transects was I gathered the drainage direction values and put them into Excel and attached vectors to each value. So it gives a value, I think it's 1 to 128, but it's exponential. And so I attached the vectors to them. So instead of looking at numbers and colors, you can actually visualize the direction that the water is flowing out of. And that's what that ended up looking like. The forest is that area there. And so the rest of it is just the rest of Palmer Park. They have a golf course there as well. Google Earth Engine provides data visualization and mass quantities of raw data, but it doesn't have the options for statistical analysis. So to account for this, I exported the data into Excel, where I could easily test how the data fits together. We were able to tell that drainage direction was influenced either by forest density or by forest health. To confirm this, more research is required, as we only looked at that specific forest patch, and we would like to look at more. In the immediate first step going on from this is going to be inverting the highlighted area so that we can look at the non-forested area in Palmer Park and pretty much just see if we can find, hopefully, that the elevation does correlate with drainage direction, since the main thing we found was drainage direction isn't directly related to elevation in the forest. So we want to find if that holds and test to see what those variables look like in the parts of Palmer Park that isn't forested. On top of that, there are countless other images that we can analyze, as the number crunching never stops. With the skills I have learned and the trends I've noticed, I am hoping to continue conducting research at University of Detroit Mercy through directed studies, and I hope that the contribution of this research can help Palmer Park, can bring actionable data to Palmer Park so that they can start looking at how to revitalize the forest. Thank you.
Hello, my name is Colin Kemmerling. I'm currently a senior at Lake Superior State. I'm in their fisheries and wildlife management program with concentration in fisheries. And for this project, Dr. Jonathan Dobeck was my mentor or my advisor. And we looked at the seasonal, how seasonal and water temperature affects lake trout diets and then their consumption of round gobies. Okay, so some of the things that might drive lake trout and round goby interaction is a decrease in like historic forage species like bloater. In recent years, that have been like alewives and smelt. But things in the Great Lakes have been changing with all the different invasive species. So we were looking at, is it possible that lake trout are starting to consume round gobies, seasonally? Some of these things that might help out these interactions are like the shifts in water temperature. And these shifts in water temperature might allow round goby and lake trout to inhabit the same like water column depth. So like I had mentioned, so the consumption of round goby, what we were trying to prove is, how does water temperature affect the consumption of round goby? So for my study, I looked at southeastern Lake Michigan at Saugatuck, South Haven, and St. Joe. And so we wanted to see when in the season are lake trout and round goby most likely to interact, and when does the diet of lake trout change? And then at what water temperatures are lake trout most likely to consume round goby, and how like thermal stratification might affect that. So my data collection started in April, and I actually just collected my last samples the 31st of July, so pretty recent. So lake trout and diet species were measured to the nearest centimeter, and lake trout were weighed to the nearest pound. The reason we didn't take weights of the diet species was just because of how degraded some of them were after sitting in the stomach. And then after, taking the weight, length of the lake trout, the stomach was then extracted and did a diet analysis on the stomach itself. And then water temperature data was collected from the various weather buoys at each point using Seagull, which is funded through Great Lakes Observation System. So the data analysis that I conducted, we looked at the percent of round goby in lake trout diet and then compared by month. So we did it individually. So whether it be April, May, June, and July as an individual, and then as a collective. So April through July. T-test we then used to see if there's a significant difference between early and late sampling times. And then we also used simple linear regression to see if there's a significant relationship of round goby to lake trout as the temperature within the water column varies. So this graph right here is just a composition of all the lake trout that were collected. So for my project, I collected 95 lake trout, and of those 95, 38 of the lake trout had identifiable species. So there was many other lake trout that had, something in their stomach, but just couldn't be identified because of how degraded the sample was. And then just to give you an idea of the size of the lake trout that I was looking at, lengths varied from 41 centimeters to 84 centimeters, and then weighed 1.1 ounce to 17 pounds. So in the graph, you can see that, lake trout predominantly consume alewives as a collective throughout the year, with goby, sculpin, and smelt mixed into their diet. But one thing that I found that was really interesting is if you look at this graph here, is that in April and May, round goby make up, 25 to 30% of lake trout diet. And then once we hit June and July, no round goby is found within their diet. It switches to almost all alewife diet. So this graph is just a graph of the t-test showing early versus late round goby consumption as a percent within lake trout. So early in the season, lake trout, their diet was comprised of roughly 25%. And then later in the season, that dropped off to 0. Now this figure can be a little bit confusing. This is the linear regression that we had done. So each of the points within the graph represent a sample date. And so there's many lake trout that are comprised within each point. And then on your x-axis, you have the water column temperature differential. And so what we really wanted to do is have on our x-axis was Smith stability, but we weren't able to find the hypsometry of Lake Michigan. So instead, we chose to take the first meter of the water column, the temperature at the first meter, and then subtract it from the hypolimnon temperature. And so as you can see, we have a downward trend. So as that temperature differential goes up, the percent of lake trout diets that were comprised of round goby goes down. And then it was really interesting that you almost have a break point at when those temperatures vary by 6 degrees, that lake trout did not consume round goby anymore. So just as, to kind of wrap things up here, lake trout predominantly consumed alewives throughout the season as a collective, but seasonally, we did find round goby within their diet. So as an assumption, looking at the temperature differential, we assume that lake trout are consuming round gobies during weekly stratified periods. And then, so our next step for, continuing on my project at Lake State is to, find the hypsometry data and calculate Smith stability, and then compare, how the lake trout diet what percent round goby are within lake trout diets at that point. And that future research in this field is extremely important, especially how the Great Lakes are changing, with climate change, whether it be water temperature or, invasive species and such. So looking at how these different factors affect, the lake trout consumption of round goby. So at this time, I'd like to give a special thanks to Michigan Sea Grant for giving me the opportunity to accept this internship, and then Seagull for sending me all the different temperature profile data that I needed, and then Best Chance Tube Guide Service, Penn Rod and Reel Guide Service, South Haven Steelheaders, and then Seahawk Fishing Sports for allowing me to collect the lake trout as they were, bringing their clients in with their catch. And I'd also like to thank Brianna Abercrombie and my parents for, going on these early morning excursions to collect data. So, thank you.
I just want to thank the interns again for the great presentations they have, and congratulations